Fluid mosaic model. You know, this is the most acceptable model of the plasma membrane. Protein and lipids are arranged in a mosaic fashion. Means, as it was suggested by Denner and Davison, that protein is not in a form of a continuous sheet, but it is arranged in a mosaic fashion. Lipids and proteins are in the semi-fluid structure. Fluidity of plasma membrane is due to phospholipids. Phospholipids of plasma membrane are rich in unsaturated fatty acids. That's why these occur as liquid. Main component of the cell membrane are phospholipids because these form the structural framework of the cell membrane. In lipid part, plasma membrane also contains cholesterol. Cholesterol is mainly found in the inner half of the membrane. Cholesterol molecules are more rigid than the phospholipids, and therefore presence of cholesterol confers stability to eukaryotic membrane. Of course, in prokaryotic cell membrane, cholesterol is absent, but it has been observed in some bacteria pentacyclic sterol-like molecules, which are referred as hupenoids, are present. Hupenoids stabilize the bacterial membrane. Now, here is the fluid mosaic model, which was suggested by Singer and Nicholson. You can see here, you have oligosaccharides, glycolipids, integral or intrinsic membrane protein, and integral and intrinsic, and you have extrinsic or peripheral membrane proteins, and you can see the cholesterol. Plasma membrane may be termed as protein icebergs in a sea of phospholipids. Integral or intrinsic proteins tightly held to the phospholipid bilayer. They also cover minute pores and account for about 70% of the total protein. Intrinsic protein form channels which provide a rapid passage to water-soluble materials across the plasma membrane. Peripheral or extrinsic protein, they account for about 20 to 30% of the total proteins. Superficially arranged on either side of the plasma membrane and can be separated easily. These proteins have enzymatic activity and they are commonly referred or called as permeases. You can see both the type of the proteins here, extrinsic proteins and intrinsic proteins or integral membrane proteins. Extrinsic proteins function as carriers for the transport of materials. Plasma membrane has approximately 30 types of enzymes, say ATPases. Plasma membrane regulates transport of materials in and outside of the cell. Plasma membrane is semi-permeable and as it allows rapid passage to water molecules. Plasma membrane is selectively permeable as it allows certain solutes to pass through and it prevents others. The size of the molecules which can pass through the plasma membrane ranges between 1 to 15 angstrom. Now you can see the facilitated diffusion through protein channels and of course the active transport. You can see, but in active transport, expenditure of energy is also involved and the source of energy is ATP. Osmosis, movement of water, that solvent across a semi-permeable membrane is called osmosis. In process of osmosis, water moves from a hypotonic solution that is of less concentration towards hypertonic, that is where it has a higher concentration solution. You can see the movement from hypotonic to hypertonic. Diffusion, movement of solute molecules from their higher concentration to lower concentration is called diffusion. Diffusion occurs in direction of the concentration gradient, so it does not involve expenditure of energy, or you can call it as passive transport. Diffusion continues till concentration of molecules becomes equal on both sides of the plasma membrane. It is called Gibbs or Donan's equilibrium. You can see the diffusion from high concentration to low concentration unless an equilibrium state is attained. Non-polar substances, which are more soluble in lipids and less soluble in water, move rapidly across plasma membrane as compared to polar substances, which are less soluble in lipids and more soluble in water. In substances of similar solubility but of different molecular weights, small molecules move more rapidly across the plasma membrane. Permeability also depends on charges of the particles. As compared to electrolytes, non-electrolytes move rapidly. Monovalent ions like sodium, potassium, chlorine move rapidly as compared to divalent ions like calcium, magnesium, and sulfate. Facilitated or mediated diffusion. This type of diffusion occurs according to concentration gradient, but it involves carrier-like permeases and translocases, etc. Active transport, or you can call it as uphill transport or metabolically active transport. In this type of transport, movement of substances, that is, transportant, takes place against their concentration gradient, and the carrier molecules which occur in the plasma membrane become attached to transportant, and a carrier transportant complex is formed. Now here you can see, in this case, the energy is obtained from the breakdown of ATP. You can see diffusion, energy is not involved. Facilitated diffusion, this comes under passive transport, and of course, expenditure of energy is involved in the active transport. At the same time, ATPase enzyme splits ATP and provides energy for the movement, that is rotation of complex. Movement of ions, that is sugars, amino acids, etc., through plasma membrane takes place by active transport. Several theories are proposed to explain the active transport through plasma membrane. Some of them are cyclic AMP theory given by Sutherland, lecithin carrier theory given by Bennett Clark, cytochrome pump theory given by Lundegard, sodium pump theory given by Hodgkin and Guinness. Pinocytosis or cell drinking. This name was given by W. H. Lewis in 1934. Ingestion of liquid by plasma membrane in form of vesicles, that is called as pinocytic vesicle or pinosome, is called pinocytosis. Here you can see the phenomena of pinocytosis. Phagocytosis or cell eating. Metcalfe proposed this term. Ingestion of solid complex materials by plasma membrane in form of vesicles, which is called as phagosome, is called phagocytosis. Pinocytosis plus phagocytosis equal to endocytosis. Exocytosis or emiocytosis or cell vomiting. Ejection of complex waste material from cell through the plasma membrane. And here you can see mitochondria, another organelle. It was discovered by Colliker in 1818, the striped muscles of insects, and he called it as cytoplasmic granules. Fleming suggested the name phyla to it. Petsium called them as sarcosome. Richard Utman called these organelles as bioplasts. The term mitochondria was suggested by Benda. Kingsbury and Hugeboom related them with the cell respiration. Pelletier worked out the ultrastructure of the mitochondria. Nash and Nash, Abzelius, and Margaret discovered DNA in mitochondria and called it as mitochondrial DNA. It is also known as organelle DNA. Mitochondria are spherical or filamentous in shape.